Okay, so this is General Biology Bio131, and this is part 2 of chapter 6. And last time we were talking about the genes, and the last thing that we talked about is how these triplets in the genes, which are called the codons or the codes, how can they translate into amino acids, and these amino acids are going to become proteins at the end. So now we're talking more about the genes, and the question is, are your genes, are you exactly what is in your genes, or there are other things that can affect the physical or behavioral traits that you can see? Obviously it should be both of them. So this will bring us to the gene regulation. Each one of the genes that we talked about, do you guys still remember the genes? Which is part of the chromosome that have a specific sequence. If you still remember this. So the gene itself, which we mentioned that the gene codes for, one gene codes for what? Huh?
What is it?
One gene codes for what? What does it code for?
A specific trait.
It can be a specific trait, but how? It codes to one protein. I mentioned that, right?
Yes.
You have the whole chromosome that contain DNA. There are genes on the DNA. We started. There are genes on the DNA, and each gene is exactly one protein. One gene translates into one protein. Inside the genes, we have these triplets. Each triplet is coding for amino acid. Is that clear? So each triplet, a code, is coding for amino acid. A gene is a sequence of these triplets that translate into amino acids. You hold them together, you put them together by a peptide bond, and you're going to make the protein. So now, which genes are going to work, which genes are not going to work? Like if you have the whole entire DNA, for example, which contain different genes, and the chromosome contain DNA, all these genes, which ones are going to work and which one are not going to work? Not all of them work all the time. It's actually being turned on and turned off. You turn on the gene, so when you turn on the gene, it's going to make a copy, codons, messenger RNA, and all this process. But if it's turned off, it's not going to do anything. Yes?
Can you give me an example of a gene that can be turned on and off?
All genes can be turned on and off. I'll give you an example of proteins, for example. If we need this specific protein, okay, collagen, I need collagen now. The gene that codes for this protein is going to be active and it's going to be on. I don't need collagen. I don't need collagen in my skin, for example, right now. I don't need it. It will be turned off, okay, and so on. So the genes can be turned on and off. Otherwise, if the genes are always on, you can get a cancer, because who's going to stop the genes, right? So you turn it on as needed, and you turn it off when you don't need it. If it is completely on, you're producing, this gene is responsible for this protein. It will make unlimited amount. Okay? These group of genes are responsible for this group of protein that make this cell. If that's on all the time, you're continuously making proteins, cells, and more cells until you end up getting a cancer, which doesn't happen because you turn it on when you need and you turn it off when you don't need it. This point clear?
Yes.
So the genes are not always on. You can turn it off and turn it on. And here is a good example that you asking about example. Here is a good example. The gene that code for lactase. Lactase is an enzyme, and the enzyme is protein in nature, if you still remember. The enzyme itself is protein in nature. So the gene that make this protein, which is the lactase enzyme. The lactase enzyme is a protein. So the lactase is going to break down what? What is it supposed to work on?
Milk.
The milk, right? What part of the milk? milk sugar. What's the name of the milk sugar? Lactose, right? Lactose. So in this case, this is an enzyme which is protein in nature. The gene that's responsible for this is on. So you have the lactase enzyme, and you're going to digest the lactose, which is the sugar of the milk. Remember this rule. Anytime you see ASE at the end of any word, it means an enzyme that breaks down something. Lactase is an enzyme that breaks down lactose. Okay? Galactase is an enzyme that breaks down galactose. Lipase is an enzyme that breaks down lipids, and so on. Anything that ends with ASE, an enzyme that breaks down something. So that's going to break down lactose, which is the sugar of the milk. But the question is, which cells need to break down the milk sugar? Like if you're drinking milk, right? You're drinking milk, and the milk is in your stomach and then go to the intestine, and then you need to break the lactose of the milk down so that you can absorb it, right? So which cells needed the lactose? It is the intestinal cells, the cells of the intestine, because when you break down the lactose, you break it down. Remember we talked about this before, monosaccharides and disaccharides and polysaccharides. Remember this? So lactose is galactose and glucose, right? If you still remember this, it's a disaccharide, consists of glucose and galactose. So when you eat it, you're drinking milk, right? The milk in your stomach and your intestine, you cannot absorb it. You cannot absorb the lactose as it is. You need to split it into the two components, which is glucose and galactose. Now you can absorb. Do you remember this? You can absorb. Things can go from your intestine to your blood only if it is small enough, which is a monosaccharide in this case. So you need to split it into glucose and galactose. Once it goes into your blood, what goes into your blood? Glucose and galactose, not lactose anymore. Are we following? Mm-hmm. It's not lactose anymore. It's glucose and galactose. So those two monosaccharides are inside your blood, and they will go to different cells. So your cells are going to utilize those two. Do you need the lactase somewhere else in your body? No. Where do you need it? You will not absorb it before you break it down. Now we do not have any lactose anymore. So why do you need lactase? Like, do I need lactase in my liver cells, for example, or my skin cells? Did you get what I mean? The lactose is a disaccharide. It consists of two parts. You cannot absorb it. You split it. Now I have the two monosaccharides, glucose and galactose. This gets into your blood. So it's in your blood now. It will go to everywhere in your body. What is going to everywhere in your body? The monosaccharide, the glucose and the galactose. How about the lactose? No more lactose.
It's already split.
It's already split. So why do you need lactose? So which cells are the only cells that need lactose? It's only the cells of the intestine, because this is the only location where you're actually facing the lactose. Did you understand this? Mm-hmm. So that tells you that the genes of lactase is in every single cell in your body, but it's turned off. You were asking about this. So the lactase, the gene for lactase in the intestinal cells is turned on. The gene of the lactase in other cells, it's turned off. Why? No need for it. Why do you want to make lactase? Did you get the idea?
Yeah.
Okay. I think we all heard about lactose intolerance, and maybe some of us have it, right? Lactose intolerance, like I can't drink the regular milk. I can drink only lactose-free milk or any other types of milks, right? We know some people have this. What is their issue? Their issue is they cannot activate the gene for lactase. That's it. An inherited condition, some people have it, and their genes cannot be turned on, simply. Okay? So you need to express the genes, turn it on and express it in the intestine only. Other than that, it is it's turned off. Okay. So this is for the gene regulation, being turned on and off. The other thing is some chromosomes not just the genes, the whole chromosome sometimes is inactive. Okay, sometimes the gene is inactive. The example for that is in mammals. Mammals are not exactly like us, and even in humans. Like if this is a female, how many X chromosomes do you have in your cells?
2.
2, right? In humans, both of them are working. In mammals, some mammals, one of them is working and the other one is inactivated. If it is inactivated, we call that the bar body. The bar body. Does that happen in humans? It's not supposed to happen, but it happened during development, during reproduction. And here is how it goes. If this is the stem cell from which we make the eggs, and this is a stem cell from which we make the sperm, okay? They are going to, we know that this is mitosis and meiosis, if you still remember, okay? So you start with 1 egg, and then you do 2, and then it's going to split and become 4. Same thing here, 2. Four sperms, okay? This is how it goes. However, what's actually happening if this is an egg is give you 2, and then these will be inactivated, and you end up having 1. You start with 1, you end up having 1.
Oh.
Okay? In males, you start with 1, you end up having 4. It's not really 4, it's millions. But I'm just giving you a small example. I'm talking about 1 only. 1 stem cell end up giving you 1 egg. 1 give you 4. Why? Because the other 3 are there but inactive. We call that the bar bodies. But this is not. This is only during the development of the ova, not in fetus or embryo or humans. It's not after fertilization at the beginning. But in mammals, it can stay there. Like you have a mammal, and if you check your cells, they can have inactive X chromosome. So that's what's happening, and let's explain it. If you ever ask yourself, like in males, you can produce millions of sperms, while the number of ova in females is limited. Do we know that? Like there is 1 only per month. But how many for males? Millions. Why? Because you start with 1, you end up having 1. You start with 1, you end up having 4. And then you split more and more and more and more. So in this case, this is called a bar body, which is inactivation of a set of those. In mammals, the X inside the cell is the one that's inactivated. Okay? So you can activate the whole thing, or you can activate the X chromosome only. If you inactivate the X chromosome, we call it X chromosome inactivation. If you inactivate the whole thing, we call that the bar body. And in this case, you're inactivating the whole. We don't call it an egg. It's supposed to be an egg, but it's inactive. So now if we are going to regulate, I mentioned only one of the regulations, which is you turn on or you turn off the genes, right? Mm-hmm. Is that the only thing? No. If that's the only thing, and if something goes wrong, and it's turned on, you will have a huge issue right away. But you have several points, like checkpoints, okay? Like if you're looking for somebody, a criminal or something on the street, you can make more than one stop, right? If he skipped this one, the next police car will pick him up. If that's not the case, the next will pick him up, right? Yes. And so on. So you have several checkpoints to make sure that you're regulating everything. The first part was the gene itself. The gene regulation, is it on or off? The second part is transcription. So the gene is already on, okay? And you are already making a copy of that gene in the form of, with transcription, what are you making? Which type of RNA?
mRNA.
M, messenger RNA. This point by itself will start, do you guys remember the video I showed you? If you wanted to make a copy messenger RNA out of DNA, we call that transcription, right? Yeah. You start by unfolding, unzipping, and then there is a primer, and then you start making that messenger RNA, right? The point where it holds of RNA, in this case the RNA polymerase, do you remember this? What's the difference between RNA polymerase and DNA polymerase?
DNA is larger.
Polymerase. I'm not talking about DNA versus RNA. I'm talking about DNA polymerase versus RNA polymerase. What are these? DNA polymerase is an enzyme that helps you to make DNA from DNA, which is replication. RNA polymerase is an enzyme that helps you to make RNA from DNA, which is called transcription. We talked about that Tuesday. Okay? We should study as we go. So what's happening here is when you start to do the transcription, and where you're going to start at the start point in which the RNA polymerase is going to start, this is also regulated. You can turn it on or turn it off. Okay? So this is another one. The third one is, or the next one is, even after you do the RNA, you can still regulate, but in a different way. The RNA, the messenger RNA that you just formed, is not perfect. It's not perfect. Because when you take a copy, like if I give you the lab manual or a textbook, and you put it on the copy machine and you make a copy, you can have a black part here and a black part here and some white on the sides, right? Mm-hmm. So that's not perfect. That's not perfect. You need it small, neat, organized, right? So here's what's happening. You make transcription, which is the messenger RNA, and then you take that copy, the newly formed copy, let's say it's like this, okay? And I have your names here, but I have no need for the white outside, right? So I'm going to cut it out, okay? And the last part in here, cut it out. You see a couple of lines, somebody's not here today, right? I will cut this. And then the remaining, which is the one that's meaningful, the one that has actual information, I will put it together again. So now I will have a clean copy that have only instructions. Yes?
So when you make a copy, there's extras?
It's not extras, it's parts that doesn't mean anything. And we call these parts introns. Introns like this. Look at this. The white, that's useless, doesn't have any information, is introns. And here we have the names. These are called the exons. Which one is good for me? Which one that have the instructions?
The ones that are signed.
The one that's, which is called exons, right? Do I need the white? Does the white have anything?
No.
This is called introns. So if I have this big copy, okay, I have exon, have information, and then a blank spot, intron. You have to cut it before it leaves. Yes?
Could they be changed exons?
No. It's introns, it's just there, it's useless. So you will just cut it. You have a question?
So is it like a damaged copy of the gene, or is it just...
It is not a damaged copy, it's just a space.
Blank?
Yes, blank. Just a space, blank.
How come they're there? What are they for?
It's just holding the pieces together. It doesn't have a real function.
Okay.
So if you look at this whole sheet here, you are going to see this where it say introns, like this is 1, this is information, right? This is 2. How about this piece in between? Useless. Cut. Between 2 and 3, blank. Cut, cut, cut. So we're going to cut all these introns and end up having exons. Put them together. This is 1. The other thing is you will have a cap on one side and a tail on other side to make a direction for this newly formed messenger RNA to know how to move out of the nucleus and go to the ribosomes and so on. So you remove the introns, you put the exons together, and then you add a cap and a tail. Is that clear? Mm-hmm. These are 3 changes that you're going to do before you have a neat, good, perfect messenger RNA that's ready to leave the nucleus. So in this part, you have the introns that you're going to cut, and all these that have the numbers. These are the exons that you need. Put them together, remove the cap and the tail. Next step, the gene is on, so you're fine. Transcription is starting, that's fine. You already made messenger RNA, and then you regulated that. You removed the introns, you rearranged and reattached the exons, you added the cap, you added the tail. And this is leaving the nucleus and go to the ribosomes. Now it's the time to translate. Do we still have a say on that, whether we translate after all of this? Yes, you still have a say on this. Should we translate or not? Should we proceed with the translation or not? You can translate and you can start. There are factors. This is a ribosome. Here is one unit and the messenger RNA. There are some factors that can come in and stop it. You can still stop it from there. Not only that, after all of this, and you already did the protein, what is the fate of the protein? You still can control that. You can break down the protein if you don't need it. You can modify if you wanted to export. So all of these steps are regulation, and you're changing something. You can change, not just yes or no. It's not only yes or no. You're making protein. Okay, it's done already. But do I have a say on that protein? You can still change it a little bit, and so on. Yes?
So that over there on the RNA, those are the head and the tail, right? The yellow?
The cap and the tail.
Okay, cap
Yes.
and tail.
The cap and the tail. Okay, so we have all these regulatory steps. Not just regulatory. We call it regulatory, but regulatory doesn't mean yes or no. Doesn't mean on and off. It means on and off. And can you change something? Like protein, it's already a protein. You didn't stop it. But can I change that protein? Can I change the nature? Can I break it down? Can I add something to it? Can I package it? Right? You have a say on all these steps. And if something of that change, it will give us different issues that we are going to talk about. So if you talk about the 3 letters, what do you call the 3 letters again?
What do you know? Triplet?
Triplet, also known as the code or codon, right? Triplet, codon, and this should be translated into protein or amino acid. Amino acid. A chain of these triplets are called gene, and this translates into one protein. Why this sound like kind of hard? The whole DNA contain group of genes. Each gene translates into one protein. Within the gene, there are these codes. Each code is translated into amino acid. When you put the amino acids together, you make a protein. Is this clear?
Yes.
Okay. So talking about these triplets, what are the mistakes that can happen? The first thing here is just one letter can make a difference. If something happened, whatever the reason is, things can change. So look at this. This is a meaningful sentence, right? The cat ate the rat, right? Now, if you just change the R, what does it mean now? Read it. The cat ate the, completely different, isn't it? Just one letter. Okay, you're changing the meaning. How about this? The cat ate. If you change, if you add like this, if you add the B, the cat ate the hat. Now it's this, okay? If you add a letter B, okay, right here, insert the B. Now the whole triplet is going to change. Look at this after adding. The cat, that doesn't have a meaning, right? Just you added one letter. Did you get the idea? Compare this to this. All what you did is CAT, right? Just put B here. It will become cab, and then the T will shift to the next one. It will become tat, and the E will shift to the next one, and so on, right? Did you notice that the whole thing now is different? So the first one you changed letter. Okay, you did not insert. You changed letter. You switched. This is scenario number 1. Scenario number 2, just adding. Everything is as it is, but just adding a letter. Scenario number 3, if you delete one of these letters, okay, like remove the C from here, this. If this is removed, it will be the ATA, right? TAT. Did you get the idea?
Yeah.
So what can we do? 3 things so far. Number 1,
Change one letter.
change letter.
Add
Number 2, add a letter.
or remove.
Number 3, remove a letter. Okay? Yes.
Is this what causes mutations?
This is the mutation.
Oh.
Okay? So this is one of the ways in which the mutation is going to happen. Yes.
How do mutations occur? Just like that.
We will talk about that. There are different reasons. Can be spontaneous, like we don't know, just happened, and it can be because of something that caused this mutation, something like carcinogenic, like a chemical compound, like radiation, like stuff like that. Okay, so I can replace, delete, or add one of the nucleotides of the triplet, and this will lead to different results depending on exactly what happened. You cannot say, or you cannot claim that just because you change something, the whole thing will become irrelevant. No, it depends on the result. Read after you changed. You changed something, then read it, and this will make the difference. Like if this is a protein, if this is a regular protein, triplet, okay? So this protein consists of serine, histidine, serine, thymine. And then you added the letter. Do you remember that pi that we were using to translate? Isn't it going to be translated into different amino acids? So if you have all different amino acids, is it the same protein that you wanted? No, it's a completely different protein depending on the result. How does it look like after you change something, after you mutate? Unfortunately, most of the time when something like that happens, most of the time, it's harmful. Will give you a bad protein that you don't need, something that's harmful to the cells. It can lead to cancer. It can lead to bad things. But there are some exceptions. What are the exceptions when mutation happen? The first exception is natural selection, evolution by natural selection. Okay, you see something changing and surviving, and something else did not change and it's going to die. Are we familiar with this in evolution? Yes. How the evolution occur and who's going to survive? Or do you know how they survive in the first place? Most of the time what happened is a mutation happened, but it was not a bad mutation. Okay, it's a good mutation, so it enabled them to survive this whatever changed, and those who couldn't survive, who couldn't have this mutation, they are going to die, and this is naturally selected to continue. Okay? Did you get my idea?
Yes.
So this can lead to natural selection, evolution by natural selection, and this is something that's not bad. Actually, those who were chosen for the natural selection are those that are going to continue, which is not something bad. On the other hand, do you remember that amino acids can have more than one code?
Yeah.
Do you remember this? Sometimes it's 2, 3, or 4. Do you remember this? 3 different codes mean the same thing. So what will happen if you change a letter, but you ended up having translated into the same amino acid? Is that an issue? It is not an issue, right? So how can mutation of the DNA happen? It can happen spontaneously, which you were asking about. Spontaneously, like, so when you say spontaneously, meaning you don't know. It's just during rearrangement one is lost for some reason that we don't know. Or it can be something due to mutagen. Mutagen is mutagen. Those are generating the mutation, or the mutation generators. Did you get where the word came from?
I got a question. See how when you go to a dentist and they do the X-ray, how much radiation does the lead that you wear?
As long as you're wearing this, you're 99% covered. and it depends on the frequency. You know how you lose your teeth? You lose your tooth when, like, the deciduous teeth. You don't like lose it all of a sudden, right? It takes long time and start to become loosen and time after time after time, and then it's going to fall. Same thing here. Usually you do not like be exposed to radiation unless it is a huge radiation, like you get a box or you get a hit. It can break your teeth, right? But if that's not the case, things are going to go gradually, right? Like, what if you are one of those who travel a lot, like once a week, using the airplane, right? And they expose you to that X-ray on a weekly basis. This can lead to cancer. Okay? How? The same idea that you're losing your teeth. It's time after time after time after time. So if I travel once every a year or 2 or 3 years, nothing much. Okay? But if you travel like on a weekly basis or bi-weekly basis and they exposed you to that radiation, it's going to hit 1 time, 2, 3, 4, 5, 10, 15, 20 times. After some time, it can actually lead to a mutation. Yes?
So, for like the X-ray, it's long-term effect?
It's a long-term, it's cumulative long-term. It takes long time. So that's why I always advise people, if you travel too much, even though it's bad to deny the X-ray, they will make a search, and it's more complicated search. They take you to the room, and you know that all that stuff, which is very inconvenient. But inconvenient is better than being exposed to that dose of X-ray. Again, if that dose of X-ray is happening on a yearly basis or something, it's not a big deal. But if it's happening on a weekly basis or something, it can lead to one of those mutations, okay? Which we call it the high energy radiation. Is that in the airport is a high energy? It's moderately high. It's not very high. The very high, you can expose to it like a mile away, and you get cancer. Like in the nuclear reactors or something, if you're even just pass by it one time only, it's over. This is very high energy radiation. But the ones that we can be exposed to, like if you're doing an X-ray, dentist, or just a regular chest X-ray, or in the airport or something, this is not a very high energy radiation. It's mild to moderate. Chemicals is one thing, like cigarette smoking, like these, you're using oil, different types of oils and stuff that can irritate, and it can lead to mutation as well. Okay, so I mentioned that it's not just because you have a mutation you will have a big issue. That's not true. It all depends on the result. What happened after the mutation? Read it, and when you read it, you will know if something is going to happen or not. So look at this one here. ACA
UGU
and UGU changed to UGU for some reason. No, I'm sorry. UGU, okay? It means cysteine, okay? What if you changed that G to C? It's the same thing. So, is there anything happened?
No.
We call that silent mutation, okay? Remember that you change just the code to another code of the same amino acid. Is that harmful?
Oh, so you remember that chart that you showed us?
Exactly. Yeah.
Okay.
It is ACG means cysteine. UGC is also the same thing. So what? What's wrong with that? It will give you just the same amino acid. You just change to another code. If that, you're lucky, because nothing is going to happen. You end up having the same amino acid. The protein will be fine. Everything is fine. We call that silent, meaning a mutation that didn't have any bad effect. It's silent. This is one. Okay. If you are making a different, if you're getting a code that's coding to a completely different amino acid, like it end up giving you serine, for example, we call that missense. You missed the amino acid that you're supposed to make. Is that okay so far? You missed. What if the change lead to a stop sign, UGA? Remember I told you about this?
Yeah.
These three means stop. Okay? This is the code. So if it is supposed to be like this, which means an amino acid, and then you change it to one of those, it can be like this, or it can be, yeah, it's the third one in this case. You ended up changing it into a stop sign. What's going to happen? You're not making the complete protein, you make part of it. Okay? And we call that the nonsense. Okay? The way I remember, you have to remember this, but the way I remember it is, it's nonsense to stop all of a sudden. It's nonsense to stop all of a sudden. This is how I remember it. Silent mutation, a mutation that's silent. It didn't make change. Missense, this is how I remember those. You missed the amino acid that you're supposed to make. Okay? It's nonsense to stop all of a sudden. Did you get the three types of mutations?
Yeah.
So it all depends on what you end up making. We call, whether addition or deletion, we call that a frame shift. Right? If you have something like this, and you put something, here is A, okay, U, and then I put U here.
So we AAUE.
Everything else will be shifted, right? So it will be like AAU, and then the A is going to the next one. It will be AUU, and then the U will follow the next one. So this is the whole frame. After that is shifted. Did you get the idea? We call that frame shifting if you're adding or deleting. But what if you are just switching? Does that lead to frame shifting?
What do you mean by that?
Switching is just like this. Does that lead to frame shifting?
So you take in one and put in one.
Yes.
No, that's gonna be a something.
Yeah, you just have this. This one is wrong, but the frame itself is the same. Like everything that follow that will stay as it is. You're not pushing it or pulling it, right?
So it's gonna be different sequence, different alphabet, say.
Yes.
Yeah, but they're not gonna lead to the.
This one only is going to change, but anything else is not going to change. Let's say after that it is G, G. It's not silent because this one is dead now.
Yeah.
Okay, so this is not really silent. You changed a letter. Okay, it depends on the result. I don't know.
So if you change even one letter.
If you change one letter, one amino acid is going to change.
Okay.
Is it going to change or not? I don't know. What if you change it and the other code for the same amino acid? I don't know. Okay? It can happen. It can be silent, it can be missing, it can be whatever, like the three that we talked about. It can be silent, it can be missing, it can be nonsense. So it all depends on this one. But it's still a mutation. And it can lead to one of the three anyway. Whether we're talking about adding, deleting, or changing, is that clear? Replacing or adding or deleting, all of three it's a mutation anyway. But this is not the point. And what the mutation leads to, it depends. When you read it, it can be silent, it can be missing, it can be nonsense. Is that clear? Which one? Anyone. But I'm making another point now. I'm talking about something else. I use frame shifting. If I change only one letter, which is replacement, this is not frame shifting. This is a frame. Okay, let's say C. Okay? Nothing changed here. The frame did not change. Those 3 will stay the same. Just this one will be affected. But if I added something,
What it's called in this case when you take.
so replace one is called replacement. Replacement mutation. Okay? What if I deleted one or inserted one? You know what's going to happen? Everything after that mutation is going to change. We call that frame shifting. So let's say I remove this. So now it will become like this. Right? And then it will become like this.
It must be in a set of 3.
Yes, it has to be 3 anyway. Okay, and so on. So everything changed after that.
Yeah.
The whole frame is shifted. We call that frame shifting. So does frame shifting happen with replacement or addition or deleting? if you understood what I mentioned.
And now the whole thing gonna be used up. Yeah.
Which is frame shifting. Frame shifting, does it happen with replacement or addition or deletion?
Addition and deleting.
Yes, addition and deleting. These two only will lead to frame shifting. Replacement does not lead to frame shifting. Okay? This is for the frame shifting. Now, cancer. If you have somebody that is diagnosed with cancer, what's cancer? It have a lot to do with what we just talked about. In our body, we have some genes that or some parts that regulate the genes, okay, and we call those proto-oncogenes. Proto-oncogenes. Onco means tumor, okay. Onco, translation, cancer or tumor. Are we following? Proto-oncogenes. What's the proto-oncogenes? It is this is part of the genes that's protecting you from cancer. If this part of the gene that's regulating the genes, if it change, the proto-oncogene will become oncogene. What's onco? Cancer. And what's gene? Gen. It's not gene, I'm sorry. Gen. Generation. It's going to generate cancers, or it will be the genes that are responsible for cancers. Okay, so proto-oncogene, this is something that's important for regulation. Are we following? What is controlling the genes? The proto-oncogenes. This is the one that's regulating the genes. This is the one that's saying stop. I mentioned that before. Go, stop, go, right? On, off, on, off. Who is doing that? The proto-oncogene. Proto-oncogene, okay? If that proto-oncogene changed, mutated, what kind of mutation? Whatever it is. But it has to be a mutation that has a meaning, or it did something bad. So now this that's controlling is not controlling anymore. We call it the oncogene. Yes?
So not all mutation of the oncogene will result in cancer, right?
Any mutation in the... No. Yeah, I got your question. You mean mutation in the proto-oncogene?
Every or just like...
Any mutation in the proto-oncogene that is changing the nature. Changing the nature. It has to be.
A last result, right?
It's changing like the frame shifting, something that's not like the silent mutation. It's not going to do anything, right? A real mutation that happened, changing the nature of that gene to the extent of losing control. Is that clear? The proto-oncogene that's supposed to be in charge, that's supposed to be controlling, that's supposed to be regulating, will lose its job. It's not regulating anymore. So genes will be on all the time. Protein, moving all the time. The cells are going to take it and reproduce more and more and more uncontrolled, and this is called cancer, and this will lead to cancer. Okay. There is a difference between benign tumor and malignant tumor. Malignant tumor is the cancer that I was talking about. Benign tumor is a little bit of a tumor. You're going beyond what you're supposed to do, but you did not lose control 100%. Okay? You are making more. Like what's normal? Here is what's normal. When I lose some of my skin cells, I should just produce skin cells enough to replace it. That's it. I lost 10, I make 10, right?
Yeah.
Isn't that what you need? That's it. I lost 1,000, I make 1,000. That's it. Yes.
So those excess cells that are produced when the oncogenes, after they produce too much, is that what when they form a tumor?
Yes. Yeah. Okay. So normally is the proto-oncogene is telling you turn on, make 100 cell, turn off, you're done. Did you get the idea?
Yeah.
I lost my some of my skin. You lose your skin cells every day. Okay, it happened anyway. So you're losing this superficial part of your skin every day. When you lose it, the proto-oncogene, the normal, is going to say turn on, make 100 cells, replace, you're done, turn off. Is that clear? Okay. Now if that proto-oncogene have a mutation, it's not a proto-oncogene anymore, it is an oncogene, which does not have a control anymore. So what's going to happen now? The cells, the gene is on, and the cells are producing. Just the 100? No.
More.
Whatever it is, 100,000. So that will lead to a bigger skin, a lump of skin, and we call that a tumor. Okay? If it goes completely out of control, we call that the cancer or malignant tumor. And if it is just a lot more than it should, we call that benign tumor. What's the difference between benign tumor and a malignant tumor? Number one, in the malignant tumor, you completely lost the control. In benign tumor, you still have a little bit of control. So even though you make a lump, you make a tumor, but it doesn't go beyond that. This is one. The other thing is, since you have zero control over these genes, the cancer cells are going to grow enough to the extent of invading the surroundings, right? You don't have a control. Like in that benign tumor, it's going to increase like this, and then it can stop. That's it, okay? This is for benign. For malignant, who is going to tell it to stop? Nothing. So it's going to continue bigger, and then start to invade the surrounding. And then it leaves the whole area and reach the blood and move to a distant spot. And we call that spread or metastasis.
What if there's a proto-oncogene that becomes an oncogene? Can you destroy that specific oncogene?
They are working on it. It's not that simple, but they are working on it. They can do, at least, here's what's happening until this point. They can do genetic mapping, and they can look and find this oncogene. If they find it, they will tell you, you are going to have something, okay? And if that's the case, they can work on it, but it's not completely controlled yet. So they are working on it. Did you understand the difference? The malignant is completely uncontrolled. The benign is uncontrolled, but to an extent. Does the benign tumor invade the surrounding? It does not. It just grow and stop. Does the benign tumor spread to distant organ, like start in the liver and go to the bone? No. This is called metastasis. Like part of the cancer detach, somehow reach the blood, go a completely different organ. This is called metastasis or spreading. And this is the difference between benign and malignant tumor. If somebody's having tumor, what are the different ways to treat it? Surgical removal. You can just remove the cancer. They can use radiation, and radiation is going to target the cells and kill them. Target the cancer cell and kill them. Unfortunately, you don't really kill all the cells. Okay, what they do is, if this is, it depends. I mean, I don't want to go like through all the details, but just to give you an idea, if this is a cancer, how can we treat it? It depends. Is it stage 1? Like, it's a cancer anyway, and if you leave it alone, it's going to do the full speed of the cancer. But right now, it's small like this. We call that primary cancer. If it gets bigger, secondary. If it invades the surrounding, we call it tertiary. If it go to a distant part, we call it quaternary, or the final or we call it metastatic part, metastatic stage. So it depends. So let's say it's stage 1, like a small cancer here. It's cancer anyway, but you cut it early. What they do is they cut and they leave something called a safe margin. Like, I see the cancer exactly here. No, you go like 3 millimeters more than that, just in case if there's a cell here or there or something. You cut with a margin, and then most of the time they do a combination of those. Give you surgery plus radiation, surgery plus chemotherapy, or all of 3, depending on the stage. But the one in common is surgery, get rid of the cells by surgery. Radiation, chemotherapy, it also get rid of the cells, or stop the cell division, slow down, disrupt the cell division using a chemical compound or radiation. Okay? What can you do to reduce cancer risk? Skin cancer, for example, is one of the most common cancers. It's very common. How does it happen? Ultraviolet radiation is a physical agent that if you are exposed to it for a longer time, it can lead to a mutation and cancer. So use a sunblock, for example. Do not be exposed to sun more than you should, especially it goes to the face more. So do not be exposed more than you should. The tanning booths, I completely, it's my personal opinion, I'm against it, because it's a different type of irradiation that darkens the skin, but it's also irritating. It can, like if you do it too many times, it can lead to the same thing. Exercise, so you have the blood moving faster and you get rid of the toxins faster. Screening to catch something early, like sometimes you have a lesion in your skin that's not malignant, it's benign, but it can be malignant. So we talked about benign, right? Benign. Malignant, it's malignant. Sometimes you have benign that can change into malignant. So if you catch it early while it's benign, get rid of it before it switch, right? So that can be a prevention of cancer. Healthy diets, like a very good example of that is if you eat a lot of fibers, that's very protective against colon cancer, for example. Colon cancer is one of the common cancers, and the food in the colon is irritating, but if you give it some fibers, vegetables and anything that have some fibers, it's protective to a good extent. Of course, not smoking, so you're not exposed to the chemicals. Yes?
Is it possible for cancer to be genetic?
Of what?
Cancer to be genetic.
Cancer to be
genetics. So basically, like my father had lung cancer, for example. Would I have a higher risk of getting lung cancer?
Oh, okay. Yeah. It's only more chances. It's not yes or no. More chances. It can be a genetic component is part of it, but the environmental component is higher. Okay. So if this is comparing one person who is smoking and the other person is not smoking, the one that's smoking is like 50 times more chances than the non-smoking. For genes, it's like 0.5, 0.05. It's just a little bit of more. So it's instead of being 1 in 100,000, it will be like 10 in 100,000 on one per thousand. Does it mean that you'll get it? Of course not. You don't know. But do you have a little bit more chances? A little bit. Okay? Because the other factor is a lot more, which is the environmental factor. In this case, it is smoking. GMO or genetically modified organisms. And when we say organisms, you're basically saying anything. It can be vegetables, fruits, it can be animals, right? Anything can be genetically modified. Basically, they take the DNA, they look at the gene, and let's play with that gene somehow to get something that's usually to try to do it for something that's more of beneficial, have something that have benefits. GMOs, these are the genetically modifying organisms. It can happen in plants, and what they do is simply, like if you ask yourself, how can they genetically modify apple, for example? Okay, how can they do that? They have an organism that's called the transgenic organism. They get that organism, it can be a virus, for example, believe it or not. They get the virus, but it's not a bad virus. They just get the virus and they play with the genes of the virus. They insert something into the virus and allow that virus to infect. They are virally infected animals, yes, but not because of they wanted the animal to be virally infected. It's because the virus will enter into the cells, and actually it will enter into the nucleus. More than that, it will merge with the chromosomes. So if you put something with the chromosomes of the virus, it will going to merge with your chromosomes, and it can give you a specific effect, whatever the effect is. Like for this example, they did a transgenic goat, that have the gene of human enzyme that's called the lysozyme. They wanted this animal to have lysozyme so they can be more resistant to issues that can happen to them. For example, so it can lead to longer life. It can lead to bigger body. It can lead to more resistant to infection. Okay? Specifically in these cases, the FDA, they used to completely block any GMOs in the beginning, and then later they said in the case of corn and soybean and cotton, what's going to happen? Like, why don't we just allow it just produce more? Like you play in the genes, so instead of producing 1, it produces 5. Okay, so you're playing in the genes is non-harmful. I hate it anyway, personally. I don't like anything that's even GMO, not even corn or whatever, even though the FDA approved it and they said that's okay. They changed only the gene that's responsible for producing more of the same. Nothing that's bad. But I don't know. I'm still against that. But they approved some of these transgenic, even they approved some transgenic fish. Why? It have a longer lifetime. It can resist harmful organisms. They said, why not? If we change this gene, they can fight for themselves. They can survive more. Okay, so it got approved. The way they can insert something is usually by plasmid, something called plasmid. Okay, the plasmid is a carrier of the genome. If you get that plasmid in the lab, and then of course everything is happening under the microscope so they can see. Okay, they get that plasmid and they insert, like this is the DNA, they put the gene in this location specifically. They want it to be here.
So is that like that when they add onto it?
Yes, but yeah, it's something close to that, but they change the sequence, something related to the character, not to the protein that's produced. So let's say they change that by changing this sequence of amino acid. They change this amino acid. If that if that amino acid change, it will change the color, for example.
Yeah.
Like if you have serine, histidine, serine again. If I change it histidine, histidine, serine, that's going to change the color from black to brown, for example. They can do that. So how the gene gets into your chromosomes or to the animals or plants chromosomes, they use something called plasmid.
So they change the bits of the DNA, and that's the proteins, RNA?
They change the DNA in a sequence that lead to changing a characteristic.
Okay.
Pharmaceutical company, they do that a lot. They, for example, they give a medication to an animal, and the animal produce like antibody, and they get that antibody and use it for humans, for example. Yes.
I heard that some of the farmers they give estrogen to cows. They produce more milk, and they're like they beef the cows up. Is that
yeah, this is not a genetic modification.
Yeah,
this is a hormonal control. It's bad. They're not supposed to do that, because the hormone itself is an inducer. So it can increase the milk, it can increase the meat and everything, but it can lead to irritation and can lead to cancer. So they shouldn't be doing that. But in this case, for the pharmaceutical company, this is completely fine, because we don't eat it or drink it or anything. We just take the chemical out of it. Okay, whatever the chemical is. It can be a serum, it can be an antibody that's produced from those animals, and so on. It can be any of those. And they do it the same way, plasmid that get into it and control the DNA. And this is the plasmid here, like this is the plasmid, okay? Which is, do you remember the plasmid? We talked about it before. Circular DNA, circular part of the chromosomes. So they get that plasmid, they cut it open, they insert the needed one, and they inject it using the whatever the way that deliver it into the cells. You go to the nucleus and take control over the nucleus, and then you give you the desired effect, whether this is a medication or not.
Is it plasmid only in bacteria?
plasmid is circular chromosome inside the bacteria. Okay, it's a circular chromosome.
Humans doesn't have.
We don't have plasmids, no. It's in bacteria. Okay, can you fill out this table? And this is the last thing that I'll talk about today.
No scientific evidence has been found that GMO pose any special risks to humans or the environment.
The pros is that in your in your textbook?
Yeah.
Can you tell me something out of your own thinking? Can you
It
think?
could be bad for human health.
It can, yeah, it can. So it can produce like tumors or something, right? What else? Can disrupt. If you like giving advantage to this one fish versus others, the others can disappear, right?
Yeah.
So you're disrupting the environment, right? Pros, you can get more products from something. You can produce some medications. You can treat some conditions. You can, if this person have a bad gene, how about if you change the gene so you don't have the issue that's going to be produced later on, right? So it can have a lot of benefits. If you have that in your manual, you can go through it. But we will stop here and we'll do the rest next time.